Good morning, good afternoon to you all. My name is Anthony Fields, and I am with my fellow colleagues at Virginia State University in our cytology class, and we will be talking to you today about calcium signaling, and I'll let each of my colleagues introduce themselves.
My name is Loretta Granum.
I'm Shantara Stewart.
My name is Katrina Eatis.
And we will begin our presentation on calcium signaling.
The first thing we're going to speak of is the purpose of calcium. Calcium serves as a universal intracellular signal which mediates various cellular processes such as cell differentiation, cell proliferation, and gene transcription. It also plays a role in developmental processes, including fertility and muscle contraction. Calcium is both an important and frequent component of signaling in cells varying from bacteria to neurons, and its regulation is a lifelong necessity. At rest, intracellular calcium levels are about 100 nanomolars. When that concentration is raised to approximately 1,000 nanomolars, the cells become activated. Due to its unique versatility, this increase in calcium is able to regulate an abundance of cellular processes. There are 4 units of the calcium signaling network. The first generates a stimulus to recruit calcium mobilizing signals. From there, an on mechanism is activated, releasing calcium in order to elevate cytoplasmic levels. Calcium is a second messenger that is responsible for triggering calcium-sensitive cellular processes. And finally, the off mechanism facilitates the movement of calcium out of the cytoplasm to reinstate the resting level of calcium.
Next, I will talk about the universality of calcium. Calcium can be a regulatory multivalent mechanism, and it is essential for a lot of our metabolic processes. Initially during fertilization, we have calcium spikes that begin fertilization and the inputs of cell development, cell differentiation, and cell proliferation. The calcium spikes trigger cellular responses, additionally also to, i.e. muscle contractions and neurotransmitter release. There are variations within the calcium spikes. When you have a frequency of calcium spikes, they are dependent upon the stimuli or the initial signaling location. For instance, the pancreas and liver can transmit a calcium wave between 1 to 60 seconds, and as far as for calcium oscillation in mitotic division, it can take up to a 24-hour period. So basically, overall, the universality of calcium highlights the frequency of calcium inputs and how it can manipulate cellular responses. This table shows some of the processes in which calcium can be released due to hormone. When certain hormones bind to their receptor, you'll see a rise in IP3, which is one of the components that triggers calcium release. As we can see, hormones such as acetylcholine, vasopressin, thrombin, antigens, peptide growth factors, and spermatozoa, you also see they have various signaling responses. As you can also see that acetylcholine, a hormone that we have as humans, can also trigger certain responses as well. So the hormone that binds to a particular location can also vary the cellular response.
Okay, this is a figure showing the calcium 2+ release of internal stores across the membrane. So these 5 different figures. The first one is A, is the intersole 1,4,5 trisphosphate receptors and the rhodine receptors that are spread across the surface of the endoplasmic reticulum or the sarcoplasmic reticulum. And the SR, it's a storage site for the bulk of the calcium, and it transports across the SR membrane, and it's managed by two molecules, which is the iodine receptor and the calcium 2+1 ATPase. And B, this is showing when there's a low level of excitability, there's a response to a weak stimulus, separating the individual channels to open to give a blip, which is the INSP3R receptors, or a quirk, which is the RYRs. And C, it's higher levels of stimulation or excitability of the groups of the INSP3Rs and the RYRs, and they open in clusters to generate a puff or a spark. A spark originally was found in cardiac cells, signifying calcium 2+ signals from a group of the RYR2 channels opening in accordance. The puffs are found in oocytes and HeLa cells, and they have diverse amplitudes indicating that there are either variable numbers of the INSP3Rs within each cluster or variable numbers of channels open within an individual cluster. And D, it's all the cells that are excitable when there's a high stimulation in the groups of clusters to make a puff or a spark that will cause them to excite the neighboring receptors by the calcium 2+ induced calcium 2+ release, causing an intracellular wave. And E, by the gap junction connecting the cells together, the waves trigger another wave which will travel through one cell to another to create an intracellular wave.
In this next section, we're going to go over the regulators of calcium entry and release. Excess calcium in the cytoplasm of cells is dangerous and can lead to cell death by apoptosis. Therefore, extracellular stimuli and regulation of these levels is critical to cell survival. There are passive transporters along the plasma membrane that facilitate the release of calcium into the cytoplasm. These transporters are receptor-operated channels, voltage-operated channels, and store-operated channels. Receptor-operated channels are also referred to as ligand-gated channels. They rely on extracellular ligands to bind to the receptor in order to open up the channel and increase cytosolic calcium. Voltage-gated channels are another set of ion channels along the plasma membrane that are activated in response to depolarization, which is the reduction of the membrane potential. Lastly, we're going to look at the store-operated channels, which we will call SOCs. They respond to the depletion of calcium in the internal stores, and these channels are responsible for providing the calcium signal to the rest of the cell. Inositol triphosphate receptors located along the endoplasmic reticulum work directly with the SOCs along the plasma membrane. In the figure, you can see that we have G protein coupled receptor. That signaling pathway is one of the two pathways that are produced to second messengers, which will eventually be delivered to the endoplasmic reticulum. This type of signaling requires receptor ligand binding in order to activate a nearby G protein, which will replace GDP for GTP on the G alpha subunit and dissociate the alpha and beta gamma subunits. The GTP-GA G alpha unit will bind to the enzyme phospholipase C, which is located along the plasma membrane. The activation of this enzyme will cleave PIP2 into IP3 and DAG, diglycerol. While diglycerol will remain in the membrane to further activate the kinase, IP3 will be released into the cytosol in order to activate IP3 receptors located along the endoplasmic reticulum. Now, the second pathway is the receptor tyrosine kinase pathway, where you have a ligand binding that causes the receptors to cluster along the plasma membrane and activate tyrosine kinases. This will initiate a process known as autophosphorylation. Phospholipase C gamma is another type of phospholipase C that is activated, and it will cleave PIP2 into the two separate molecules, DAG and IP3. As with the G protein coupled receptor pathway, the IP3 will remain in the cytosol to activate the IP3 receptor in the ER and facilitate the release of calcium ions into the cytosol. So next we're gonna look at the release of calcium from internal stores into the cytoplasm, which mainly occurs through IP3 receptors and ryanodine receptors, which are located along the endoplasmic or sacroplasmic reticulum membranes. IP3R is a universal calcium release channel that is located in the endoplasmic reticulum. It is activated by an inositol phospholipid calcium pathway second messenger, which we mentioned earlier as IP3. Ligand binding will open the channel and release calcium into the cytoplasm. It is a joint effort between the IP3 and calcium levels that opens the channel. Ryanodine receptors are also calcium release channels located in the sacroplasmic reticulum of all excitable cells. These cells include cardiac, skeletal muscles, and neurons. Calcium is the principal activator of these channels, and its ability to activate the receptor is regulated by another second messenger known as cyclic ADP ribose. The increase in calcium ion concentration causes the channel to open and release ions into the cytosol. This is a calcium-induced calcium release mechanism. It's a positive feedback mechanism. It is important to note that this applies to both IP3Rs and RYRs to enhance the signal until the calcium level rises above a certain level. And then the negative feedback will cause this process to end. In order to decrease elevated cytosolic calcium levels, they require the use of active transporters located along the ER, SR, and the plasma membrane to move calcium ions out of the cytoplasm. Two ATPases that maintain the calcium concentration are SERCAs and PMCAs. Those in the plasma membrane are responsible for calcium transportation out of the cell. They are plasma membrane calcium ATPases. And those in the endoplasmic or sacroplasmic reticulum provide calcium transportation into the lumen of the ER, and they are referred to as sacroendoplasmic reticulum ATPases. Both of which tend to have high affinities and lower transport rates in response to high cytosolic calcium levels, and they hydrolyze ATP into ADP in order to facilitate this process. Mitochondria can also regulate calcium transport into the mitochondrial matrix through what is called a uniporter. So it is the mitochondria which releases protons to create the electrochemical gradient that will synthesize ATP. And this gradient facilitates calcium uptake, which is usually more efficient when the mitochondria is in close proximity to calcium-releasing channels. The calcium is taken up by the the calcium that is taken up by the mitochondria is supplied by the endoplasmic reticulum. And lastly, along the plasma membrane, we see sodium-calcium exchangers, which reduce the cytosolic calcium levels. They do this by exchanging sodium ions for calcium. This image just gives us an overview of the calcium ions that are responsible for increasing cytoplasmic calcium concentration. They are in red. That's where you'll see your store-operated channels, your ligand-gated channels, and your voltage channels, along with your IP3R and RYI receptors on the endoplasmic reticulum. Those that are blue, they deplete the cytoplasmic calcium concentration by transporting calcium out of the cell or into the internal stores.
I'm going to talk about calcium binding proteins and buffers. Calcium signaling in each cell is shaped by the sundry of proteins included in each cell. This includes calcium channels, transporters pumps, and calcium buffers. The role of calcium as a ubiquitous second messenger depends greatly on a variance of calcium binding proteins. Calcium binding proteins control calcium concentration in cytosol. They also act as transport across cell membranes in a great number of cell functions. And they also act as a modulated sensor for decoding signals. There are three types of calcium binding proteins. There are intercellular calcium binding proteins that contain EF-hand domains, which are parvalbumin, calmodium, S100 protein, calcineurin, and also there are intracellular calcium binding proteins lacking the EF hand domain within the calcium binding stores, where annexins, proteins with C2 domain, and protein kinase C. The third type of calcium binding protein is the extracellular calcium binding protein, classified by calcium binding structures, and these are classified in 6 groups. That is the EF hand domains, the EGF-like domains, the gamma carboxyl glutamate acid-rich domains, gadherin domains, the calcium-dependent C-type lectin domains, and also the calcium binding pockets of family domains with GPCR. Calcium buffers are binding proteins that act as a binder and intracellular signals. They are used in regulation and signaling, along with maintaining homeostasis and calcium signaling. Examples of calcium buffers include both alpha and beta isoforms of paraalbumin, calbindin D9K, calbindin D28K, and calterin.
So, as we found previously, that calcium has the ability to affect a multitude of processes, and this table actually shows some of the calcium-sensitive processes. You see that if you have calcineurin, you can have various transcription factors that can lead to muscle contractions. You also can see that you have proliferation, fertilization, learning, and memory. Also, calcium, based on what we learned in the previous figures, that it has the ability for transactivation. So you can have crosstalk between one receptor and activation downstream for another receptor. And you also have voltage-gated calcium channels, which deal with excitability in neurons, and you can also have inhibitory responses as well. So calcium is really essential for like I said, cell proliferation, and also can lead to cytotic division. And calcium is also highly toxic to the cell, so it can also lead to apoptosis. So now that we understand those things, we see back what I said about the transactivation. Either you can have the RTK cell surface receptor or the G protein coupled receptor, and you can still have phospholipase C activation, which will cleave the second messenger DAG and IP3. And IP3 will go on to activate IP3 receptors located on the ER that can also lead to calcium release if all the intracellular cytosolic calcium is gone. And calcium has also the ability to go on to activate PKC, and which is a protein kinase for calcium, and activate other molecules inside the cell. So calcium can induce calcium release if all the calcium in the cell is depleted. And calcium can also be released in the ER. And also there are vesicles connected to the plasma membrane, which is the SOCs, that can also release calcium when the cytosolic concentration of calcium has also been lowered. And we understand that IP3 precedes the rise in cytosolic calcium concentration release.
Next, we're going to talk about the on mechanisms feedback. So initially, the signal for calcium release from the SR begins with a depolarization of the cell membrane, which travels into a specialized invagination in the T tubule. There, voltage-sensitive calcium channels, known as the L-type channels or the dihydropyrimidine receptor opens and allows small amounts of calcium into the cell. This does not raise intracellular calcium concentrations sufficiently for contraction. Rather, the primary role of these L-type calcium channels is to open the RYRs either singly or as small groups. So the calcium 2+ on mechanisms include channels located on the plasma membrane, which regulate the supply of the calcium 2 from the extracellular space, and the channels on the endoplasmic reticulum and the sarcoplasmic reticulum, which release limited intracellular calcium 2+ stores. These on mechanisms depend on the calcium 2+ channels controlling the entry of the external calcium 2+ or the release of the calcium 2+ internal stores. So these voltage operated channels are plasma membrane ion channels that are activated by membrane depolarization. So in the muscle, there's a skeletal muscle, and it's evidence for a physical interaction between the L-type channels and the RYR1 across a small gap between the plasma membrane and the adjacent SR membrane. So we have the L-type voltage operated channels, and it's responsible for the excitation contraction coupling of the skeletal, smooth, and cardiac muscle for aldosterone secretion in the endocrine cells of the adrenal cortex. After the membrane is depolarized, it causes a conformational change, directing coming from the RYR1, then releasing calcium 2+ from the sarcoplasmic reticulum. In the cardiac muscle, the L-type channels facilitate transport of small amounts of extracellular calcium, which induce nearby RYR2 channels to open by a mechanism called calcium-induced calcium release. In neurons, the N and the P/Q type voltage operating channels occur at the synaptic ends, triggering the release of neurotransmitters. So the N-type voltage operated channels, they are calcium channels found primarily at the presynaptic terminals and involved in neural transmission. The P/Q type voltage gated operated channels require strong depolarization in order to be activated. They're found on the axon terminals as well as the soma-dendritic areas of the neurons within the central and peripheral nervous system. And the L-type voltage channels are on the cell body and proximal dendrites, giving the calcium 2+ signals to influence gene activation. For kinetic filters, they have a response to small depolarizations at any synaptic spine. The ROCs are receptor-operated calcium 2+ channels comprised of a range of structurally and functionally diverse channels that are prevalent on secreting cells and nerve terminals. They're activated by the bind of the agonist to an extracellular domain of the channel. For the pancreas, they have non-excitable cells. The RYRs and the INSP3Rs work together for regulation of fluid and secretion. The acetylcholine and the cholecystokinin occur with the receptor INSP3R. The cholecystokinin also occur through the NAADP and the CADPR. There's a possibility that the NAADP can cause a release of calcium 2+ from the receptor RYR. For the sensitive processes, when there's a signal, the sensitive process is converted into a cellular response. So these sensors are the TNC, which is the tropin C, that binds to the calcium that triggers skeletal muscle contractions, causing the calcium to be sequestered into the SR. And the CAM, the calmodian, it leads to a rearrangement of the protein that allows it to bind and allosterically regulate target molecules such as the calcium calmodian dependent kinases. CAM is also mobile within the cells and can associate with its targets after binding calcium. However, in some cases, pre-bound to its target and provides rapid control. So these off feedback mechanisms, it's when the mitochondria isolates the calcium 2 when the signal is being developed and it's recovered. It's able to collect a lot of loads of calcium 2, and the matrix contains buffers, so it'll regulate how high and low the concentration is. Off mechanisms is employed by cells to remove the calcium 2+ from cytoplasm. These include calcium 2+ ATPases on the plasma membrane and the endoplasmic reticulum or the sarcoplasmic reticulum, in addition to exchangers that utilize gradients of other ions to provide the energy to transport calcium 2+ out of the cell. Occasionally, some of the off mechanisms contribute to cytosolic calcium 2+ increases, like slippage of the calcium 2+ through calcium ATPases and the exchange of calcium and sodium. Low conductor states of the pore connect reversibly, allowing mitochondria to become excitable, and it can contribute to the generation of calcium 2+ waves. Or it can have an in reverse high conductance state of the PTP, and it's a mark effect on the mitochondrion, and it collapses the transmembrane potential and leads to release of the cytochrome C and initiation of apoptosis of the calcium.
There are different types of calcium signals: transient, waves, and oscillations. Calcium signaling pathways can be lethal and require encoding and decoding. Calcium as a second messenger requires adequate stimuli and concentrations. A rapid elevation in concentration then oscillates and decreases lead to activation, termination, and modulation. Waves are specific forms of excitability, and they work in two pathways. They are direct in communication between cytosol through gap junctions, or they go through glial transmitters, which activate membrane receptors on nearby cells. Based on transient rise, oscillation, and cytoplasmic calcium concentration, they go through calcium modulates the IMP3 receptors in calcium. The EF hand binds to the in a CAM pathway. It goes Ca2+ goes under a conformational change, and then exposes hydrophobic residues that facilitate interactions of Ca2+ sensors. CAM kinases and CAM kinase 1 act as a decoder and propose decoders of Ca2 oscillation. Autophosphorylation takes place, but the biochemistry of oscillation is not known. Once CAM2 is activated, it will stay activated for several minutes after Ca2 levels have been returned to the base level, and CAM2 can integrate the Ca2 transients over time in an oscillating signal.
And yes, so now we will go on to cell specificity. And as we said before, depending on the waves, the quirk, the sparks, the blips, they can also have various effects in different cell types. As you see here, you can have voltage activation in the L type for the ryanodine receptor and skeletal muscles that can also go through to the SR to lead to calcium release that can go on to lead up to muscle contraction. And as far as for the neurons, you have the excitability, you have the glutamate receptors, and then you also go through the process of exocytosis as calcium binds to, is engulfed inside vesicles, and that's how it's released inside the cell. That can lead on to synaptic effects, and you can also have gene transcription, or you can have learning and memory. So basically calcium has the ability to vary its function depending upon the cell type. And as far as the pancreatic is where we have most of our hormones that trigger insulin. You have acetylcholine, a hormone that will combine to the receptor for the for the G protein coupled receptor, and it leads on to the PLC activation that will then have the release of IP3 that goes to the ER, and you have the secretion of fluids or other enzyme secretion. And so we basically see that depending upon the cell type, calcium has the objective or the ability to manipulate cell response depending on the signal. And even if you have a similar ligand, it doesn't always have to have the necessary response. And that will conclude our presentation today. Thank you all for listening, and enjoy it.